Okay, well, I'd like to welcome everybody to our second IMAG MSM working group meeting of April 1st. Thank you, everybody, who joined us at 11 a.m. this morning. Today, we will be following our usual meeting structure. That is, we'll have two brief talks and then an opportunity for discussion following the main meeting. So I hope people will be able to stick around to join our speakers for more informal discussion. I need to remind everybody that the meeting is being live streamed on YouTube, and the recordings of the meeting will be made available on YouTube. As usual, the people involved, myself, James Glazier, Indiana University, co-lead with Reinhard Laubenbacher, and helpfully supported by Jim Sluka and Bruce Shapiro, whom you've all communicated with. Anything technical or issues that you'd like to address, please take it up with any of us. We're always available to discuss. As usual, we also have to remind you that we have a Slack channel, we have a Twitter channel. There are the IMAG MSM Wiki pages. There is now an IMAG LinkedIn page. And we have a YouTube channel where all of the previously recorded meetings are available. We'd very much appreciate your help curating and improving the content for these communication channels. They only work if we use them. And curation is time consuming. And we hope people, speakers in particular, are encouraged to share their own talks so that the material that's available is used. Starting last week, we had the opportunity to do some short announcements. 1 minute. If you need to do a slide, that's okay. You don't need to have a slide. And so I quickly call to see if there are any announcements that people would like to make. Does anyone have any announcements that they would like to make today? We have another opportunity after the main meeting as well. We had made a comment last week that there were some requests for information on how useful this working group was for people. In particular, if you've made new research contacts, collaborations that this working group has helped you establish, that's an important metric of our success. That could be writing a paper with somebody, writing grants with somebody, preparing resources, or simply extensive discussions and scientific ideas. If you can let us know, especially send that on to Bruce, that's very helpful for us to document the functioning and utility of the working group. It helps us tune the working group. For upcoming meetings, next week we have James Moore and Denise Kirshner speaking. April 15th, Winston Guerrera and Vivek Shenoy. April 22nd, Deborah Peters, and you can see the schedule going forward. We have some people booked already into May. We really welcome your suggestions for future speakers. And as I always say, because these seminars are very, very short, people are welcome to come back and to speak again about material they didn't get to cover in the first time. We understand that this 15-minute format is a teaser. Sometimes it's frustrating to try to convey all of the interesting work you're doing in 15 minutes, and we're very happy to have people come back and speak again. As rules of the meeting, we'd appreciate it if you're not speaking, if you would mute your microphone so that we can reduce background noise. During the meeting proper, we don't have that much time for questions, so we especially appreciate questions and ideas that are high level and also from the people who don't speak all the time, because there are a few of us who tend to speak a lot. And now we have the breakout rooms for more in-depth discussion afterwards. And I will generally give a 5-minute warning if it looks like people might be overrunning their time so that we can stay on schedule. So please don't be, our speakers, please don't be offended if I simply break in and say 5 minutes. And with that, people know who are regulars that I don't do an elaborate introduction. I don't want to take any more time away from our first speaker. Our first speaker is John Yin from University of Wisconsin-Madison. He's going to be talking to us about the kinetics of viral growth in cells. So I will let you have the screen share, and please take it away.
Thanks for the opportunity. It's a real pleasure to be here and share some of our interests. So I'll start off with my slide sharing. Hopefully you can see this very busy opening slide. Don't worry, they're not all like this. This is just a little bit of an overview of sort of things we're interested in or have been interested in over the last 20, 30 some years. Quantitative virology, in a word. And we've done work with virus counting. This is useful for estimating particle to PFU ratios. We've done a number of studies in single cell kinetics of virus production, showing the high diversity that one can see from both the kinetics as well as the one-step growth behavior. Set up a few real-time systems for dual color gene expression, either by virus host, I'll show an example of that, or by virus and virus mutant. So you can look at the ecological dynamics, either in well-mixed systems or in spatial systems, where green and red here represent a couple of different kinds of virus strains. I actually started off my career doing spatially resolved evolution, showing how viruses growing in a single plaque could expand and evolve. This is a DNA profile of a loss of a gene in a particular kind of environment. And then we've also been very interested in ways that viruses can spread under the presence of fluid flows as well as diffusion. Here are some dishes where there's a little bit of fluid flow going radially outward, and this facilitates the viral spread. And more recently, this is a continuing interest of ours in defective interfering virus-like particles, which seem to be arising ever more frequently in clinical specimens. And these may play a role in activation of innate immunity as well as inhibition of virus growth. And we're also very interested in making viruses for vaccines. And I highlight here Huicheng Shi, who is in the audience. He has done some nice work on Zika virus, very high resolution, multi-samples over multiple cycles. So this is comparing a couple of strains, the African and Asian lineages. So that's a very brief aspect, focusing actually on the measures. And I'll talk a little bit about models then. So we are interested here. I am somehow not progressing on my screen. So let me see if this works. Okay, good. So title of the talk is Kinetics of Virus Growth in Cells. I've got a small lab, 3 graduate students underlined there with a lot of undergraduates, sorry. Graduate students are underlined, undergraduates are other names. Various collaborators at University of Wisconsin and elsewhere. And then I'm grateful for support from NSF and NIH. So we have a number of motivations for studying viruses. Of course, they cause many human diseases, and I should add to that list COVID-19. But there are also other reasons for studying viruses, and that's because they encode the smallest genomes. So if you are interested in how genomes become organisms, you can think of the viral growth as a toy model of development. So virus in a given environment then defines a developmental process. And of course, we have a lot of information about viruses. They've been well characterized not only from genome sequences, but we know something about the way the genes are regulated, how they function, and how they interact. There are short generation times that enable us to do quantitative kinetic experiments on a graduate student friendly timescale. So this is another reason for studying viruses. It's nice to try to get on the same page comparing viruses with humans. So this is a plot of number of proteins encoded by the genome and the genome size, where I plotted on the diagonal here 1 protein per kilobase. humans fall way off this curve, as do many higher organisms. But if we extend this curve to the lower area using mycoplasma as a point of reference, mycoplasma as the point of reference, and all these smaller entities here are viruses, they pack nicely against this curve, highlighting that they tend to be quite efficient in their coding. A lot of the coding that's viral information is actually encoding proteins. So all viruses have in common that they must make protein. So here are various different flavors of viruses, from double-stranded DNA all the way through double-stranded DNA that go through reverse transcription, and various flavors in between. There's SARS coronavirus. I put some stars here on ones where there are kinetic models of growth available. This needs to still be updated because there have been a few more that have come online since this review paper. So let's look at the intracellular growth. Here's a toy version of how we think about modeling. We and the broader community interested in intracellular kinetics of virus growth. You've got a free virus particle that encounters a cell, and various things happen inside when it releases its genome. The genome gets processed to make messages, proteins, viral capsids, more genomes. These get packaged, and free virus is released. So the process of building a model entails going into the literature, translating the biology into the simplest kinds of equations we can possibly imagine to try to describe each step, from initial binding to a receptor, to entry, to processes such as transcription. This is a sort of generic equation for how a given message might be transcribed based on genome, and its presence will also depend on decay. And the protein that corresponds to that message then also depends on the level of the message, how fast it's translated, whether or not the protein gets assembled into other complexes, and also protein decay. So that gives you a little bit of a flavor of the sorts of modeling. And ultimately, in the bottom here, we're producing free viruses. So you'd like to be able to predict how long it'll take and how much you'll make. In summary, here's a process of taking the molecular data from sequences, mechanisms, and gene functions. These feed into the mathematical models, typically coupled ODEs, sometimes algebraic equations, and we've got initial conditions, typically bound binding of the virus to its receptor. And then what you calculate by integrating forward are the various intracellular components, and ultimately, in the red curve here, the progeny virus, where there's a delay time, that how long you need to wait until you see the progeny, and based on the resources, also some level of the progeny that are coming out of the cells. So this process of some version of this has been going on for quite a while, I'd say over 50 years, where I've highlighted in boxes here some of the models that we contributed to, one for phage T7 in 1997, one for HIV in 99, and then vesicular stomatitis virus in 2006. I think it's fun to look back at the very earliest one, this paper in 1971, with Baracelli as the first author. This was virus genetic theory testing by data processing machines, something we call today computers. So this was on some of the earliest computers that had transistors rather than vacuum tubes. So when they were at Institute for Advanced Study, during the day, they were playing war games. During the evening, Baracelli would get in on this computer and do his virus models. There's a nice story of this in Turing's Cathedral by George Dyson. So we and others have developed these models. This slide focuses on works of our own to examine antiviral strategies, to look at how one might infer mechanisms from transcriptome proteome patterns, and how study genetic interactions, epistasis, these sorts of models, one can begin to simulate mutations and interactions, even explore genome design. And nature versus nurture, this is the nature is the genome of the virus versus the nurture, what's the host cell providing in terms of resources for growth. So these are largely reviewed in this 2018 paper. I'd like to take the remaining time to very quickly go over 3 questions that I think might be fun for this audience. How diverse are single cells when infected by single virus particles? Well, you can infect cells with a recombinant virus that carries a GFP. If you do this at low multiplicity, 0.01, then cells that are lighted up, you can pretty much guarantee are infected by a single virus particle. If you isolate those cells and look at the yields from then on the x-axis you have a broad diversity of yields from 10 to 500 here up to 7,500 or 8,000. So about a 300-fold variability in the yield of from these viruses. This is from 134 cells. If you isolate 8 cells and grow them up from the high yield ones, you find they all grow like the average. So there's no genetics in this, at least. And then if you isolate 7 of the low producers, you find that 5 also grow like the average, but 2 grow low. So from this kind of study, you can see that the genetic variability likely plays out in about 10 to 15%, while the rest seems to be from other factors. What are other factors? They might be actually different places in the cell cycle, where you can see that if we grow up the virus in early S phase, we get up about 1,300 viruses per cell. But if we go to G2M, then we can get almost 9,000 virus particles per cell. So there's a 6-fold difference between low and high here. Whereas if it were a purely volumetric difference, then you might expect perhaps a 2-fold difference. So there's something special about G2M. Second question, how might we visualize and measure the dynamics of virus cell interaction? Well, we've wired up cells so that the virus comes in and brings in red. And we've also got a system where the cells can be wired up such that interferon-sensitive genes will light up green. And we have a particular mutant that will bring in a red fluorescent protein. So in essence, if this mutant gets into a cell, you can get both red and green. And if we run that experiment, here is at 3.5 hours post-infection, you're seeing a bunch of blue nuclei. But if I let her rip on the movie, then you can see cells light up various colors of red and green. It's a mixture. You even see a little cell running around here before it apoptoses. So if you sit on these individual cells, you can quantify the level of viral activity from red fluorescent protein and the level of host response. This is an interferon-sensitive gene by its GFP. So when you let this rip, you can look at how each individual cell is lighting up and moving across this. This is a dynamic, then essentially a cartoon version of the movie. And you see the blue dot is our population average, but what you also see is a huge amount of heterogeneity, both in where you end up and as well as the path that you take. So that's a huge challenge, I think, for modelers. There is also opportunity in exploiting such variability to also gain insights, and I'd be happy to talk about that in the question session. Final question, how fast does a virus grow? Well, it depends on the virus. So a student went into the literature and looked at 101 virus cell pairs spanning diversity of life. This is bacteria, archaea, and eukarya, and pulled out one-step growth curves. We analyzed the one-step growth curves. And if you look at various parameters pulled out of these, like the infection burst size, the delay time, cell volume, cell doubling time, you can look for correlations. And what comes out is that, well, you can look at cells that might be over 100,000 or so variation in size. There's maybe 100-fold variation in burst size. So there's very little correlation between the cell volume and the amount of virus that they make. However, if you look at the doubling time of the cell, so a longer doubling time, slower cells out here, and the delay time of the virus, you get this very nice kind of stronger correlation. This is with 100 and 101 pairs. And this suggests that the question should perhaps not be how fast does a virus grow, but how long does a host cell persist when it's been infected? Same cell infected by different viruses may show very similar sorts of growth dynamics. So that's all I had to say. I'd love to have colleagues visit at Wisconsin or send your best students here. We have some nice grad programs in engineering and biophysics, and I'd be happy to take any questions. Thank you.
Well, thank you very much. That was a delightful talk and an immense amount of material in a very short amount of time.
I tried.
If it's okay, okay, we'll ask we'll have one question now, and then I think we'll go through the second talk and then have the questions together, if that's okay.
Sure.
Especially since the talks are in some ways very closely related, I think that may work well. Jacob, did you have an urgent question? Okay, go ahead.
Not so urgent, but I'm seeing that he's showing all sorts of movies to expand whatever he's trying to talk about. My question is related to something we're doing here in the dissemination subgroup. Will a format like a journal that publishes interactive visualizations be helpful for you? Will you publish in something like this?
I guess I would have to think about it, because I have been approached by, I think that some of these journals that emphasize the methodology, where they can videotape you. And I guess the reservations I've had is there is an additional cost for that. So it is more expensive to publish for such journals. There's more overhead. So I guess I would have to think about it, but I think there is value certainly in other ways of conveying what we do and conveying the nature of our results. So I was sad that we couldn't show those sorts of animations, and the paper itself is, you look at it and say, it's much less kind of impactful in that respect. We do go into the quantitative analysis though on both of the colors and how they are relating to each other.
You gave a good answer, and I'm not talking about movies. We're talking about actual interaction, but never mind. Thank you very much. You actually gave the exact answer I was looking for.
Thank you for your question.
Okay, thank you. And we'll move on to our second talk, which is Ron Milo. And he's going to be telling us about counting SARS-CoV-2 virions in an infected person. So we have a theme of viral quantification today, which is something we haven't heard a lot about in our previous talks. So I very much appreciate the willingness of our speakers to come and tell us about it. Take it away.
Okay.
Please.
Hi, everyone. Great to be here and to follow up on John's presentation, which indeed I think ties together. I'll be sharing my presentation in a second. I'll also say that everything I'll be presenting, I'm happy to be in this forum because I'm stepping beyond my comfort zone in some of the subjects. And I think in that respect, we'll be love to get feedback. And I've added in the chat the link to our paper draft. And so if anybody finds this intriguing, you're welcome to have a look. And I gave open edit permission, so you can put comments, give suggestions, whatever you think could make the science better. Okay. This is John. And just 1 second, I wanted to do something else. Just 1 second. I have one problem with sharing my screen. Let me try and see if I can solve this problem now. Okay. So I'll be talking about the total number and mass of SARS-CoV-2 virions, and this is work together with members of my lab, some of them joining here in the session, and with the group of Rob Phillips at Caltech, whom I've been working on for a while. And some of the things we've been interested in, so this might sound like a bit crazy to try and give values for, say, the total number of SARS-CoV-2 around the world. I'll only say that we've been doing such crazy things in the past, and I think it's an interesting thing to try and engage in. You can see some examples. I wouldn't get deep into them, but if anybody's interested, early on in the pandemic, we were trying to collect the available numbers in the literature on COVID-19. So that's something you can have a look at. And recently, we've been looking at cellular turnover in the human body, things about the increase in human-made mass around the world, trying to make a census of biomass distribution on Earth, revisit the numbers of human and bacterial cells, and all of that together from a quantitative perspective is part of a book that Rob Phillips and I wrote about cell biology by the numbers. All of that is just to give a sense of where I'm coming from. So, I'm interested in viruses, but my expertise lies in something else. And in that respect, I think you could probably help me a lot in this presentation. And so, given the 15 minutes that I have now remaining somewhat less, let me just tell you first what are the questions that we try to answer. And even if I won't cover all of them very deeply, at least the questions that also give you the key answers that we arrived at. So one is, how many SARS-CoV-2 are in an affected person? Next is, what fraction of the host cells, or as you'll see, we're mostly focusing on lung cells as the interesting one, are infected? What is the stoichiometry between neutralizing antibodies and the spike proteins? What is the total mass of virions in a person that's infected? And in total, in all of humanity, how many mutations are required during an infection? Say from the moment of infection until you're infecting another person? And how often does each possible base permutation arise and propagate? Say, for example, if you're interested in, say, this within a variant of concern of the mutation in specific mutation in 484, how often do you expect such a thing to arise in the population? And I'll also say that if you want, you can write comments and throughout in the chat. And if I won't be able to talk and answer, I think Inon Baron, who's from my team, might be able to answer in real time. So because I only have 15 minutes, let me give you the answers already now, even though it's a bit of the fun of deriving, but at least I won't keep you without the answer. So we found about 10 to the 9 to 10 to the 11 RNA copies around peak infection. That's the number of virions, if you like. And that translates into about 10 to the 5 to 10 to the 7 infectious units. And this conversion is also something I'll be mentioning in a second. If we're talking about the whole cells that are infected, even during peak infection, we find that the vast majority of potential host cells are not infected. So only about 1 in a million is infected. And I'll show you how we arrived at that. The stoichiometry between neutralizing antibodies and spike proteins is such that there's an overabundance of about 10 to the 3 to 10 to the 5 neutralizing antibodies per spike protein. The total mass of virions in a person is about 0.1 milligram maximum, 1 to 100. And within all of humanity, if you like, there's about 1 kilogram that's causing civilization to be, at a very challenging time. So that's overall at a snapshot in time. You can also ask about the cumulative mass of virions throughout the pandemic, and we've been calculating that in the link that I've shared with you in the chat. About mutations, we find there's about half a mutation per infection. That's from the moment of infection to the next infection. There'll be a change in about in every 2 such generations, there'll be about 1 mutation. That translates into 3 mutations per month, or 10 to the minus 3 mutations per base per year, which is in line with values known in the literature. And if you look at a specific mutation, such as the ones arising in all sorts of variants of concern, we find they appear every about 200,000 infections that transmitted from a person after it arises novo in that person to another person, which means that given the rate of cases globally, we have that several times appearing every day, every possible base mutation. So that shows you about the spanning of different mutations. Okay, that might have been a bit a lot to comprehend, but let me try now go a bit more deeply into things. First of all, when we're talking about the number of things here, we have to differentiate between, say, genome copies, which are usually measured, through CT or the, real-time PCR, and we're making the assumptions that this translates into what you can call a virion, meaning one genome copy would be one virion. Although this is something that we think would be very interesting to try and quantify better, because there could be also some naked RNA, like what is the fraction of naked RNA, I think is an interesting open question. And then at the other extreme, there's the infectious units, which are measured by units such as the TCID50, kind of like analogous to the plaque forming unit. Those who are coming from virology know it better. And this really relates into what successful virions would be infecting cells in future infection cycles. And these two values are quite different. There's a ratio of about 10 to the 4 we find from different sources, and therefore I'll be reporting both. And so if we're talking about how many virions are in a host, you can look at the different tissues where you tend to find them, in the trachea, in tonsils, in lymph nodes, in the lungs, in the digestive system. In each one, you could look at what is known. This is mostly from rhesus monkeys, because there aren't good data at that resolution in humans. And it's an interesting question how similar or different these would be. And you can find for each one what is the range. In terms of order of magnitude, obviously there's a lot of variation, but you can see here about 100-fold variation that has been reported. In terms of RNA copies per gram or infectious units per gram, you can multiply that by the tissue mass, again, order of magnitude of the human tissue mass of that tissue, and then you get an estimated number. And you can see that lungs are both dominant in terms of having about an order of magnitude or more concentration of infectious particles, and in terms of the tissue mass. And when you multiply that, they become by far 3 orders of magnitude or 2 orders of magnitude at least dominant, and therefore most of the sheer number is residing in the lungs. And we concluded there's about 1 to 100 billion RNA copies per person, and around 0.1 million to 10 million infectious units per person. Only a small fraction, I want to say, of the potential host cells are infected. In order to derive that, you can go through all the different stages here. I think I'll be running out of time, so I won't go very deeply into this, but this would be publicly available and also is available, as I said, on the version that I put on the cloud. But basically, you go through all the relevant multiplications. There's nothing really complicated here.
I was going to say you're actually ahead of schedule, so if you want to take an extra minute or two, it's okay. Please.
Okay, I have a lot more to cover. So how many minutes do I have left?
I guess, okay, if you have a lot more to cover, I guess you're 10 minutes in, so you still have 10 minutes doesn't seem like a lot of time, and I'm taking up your time, so I should let you talk.
Okay, so it's hard in real time and and I want to show you more interesting things, I think, still. So I'll only say there's you can do step by step the calculation. You can hopefully, if you didn't take a share screen, like the snapshot of the screen, you could just look at what I've shared with you, and you'll find all the information. But from this derivation, we find that there's way more lung cells than the cells getting infected, and therefore we get to this situation. Similarly, you can do the analysis on the number of viral particles and how they're outnumbered by the neutralizing antibodies. So for that, you take the concentration of antibodies in serum and what fraction of them are actually neutralizing versus SARS-CoV-2, and you get about 10 to the 12 neutralizing antibodies per ml. and that in comparison to the number of spike proteins per ml in the relevant tissues, and you find again several orders of magnitude mismatch or larger number. But you can also rationalize or motivate why this has to be the case in order to be effective based on the affinity of the binding, and that is again in this manuscript that I've shared with you. This thing about the overall mass that I've mentioned of about 1 kilogram, that's kind of straightforward. If you take the number of viral particles, you look at peak infection at this value of about 1 billion to 100 billion, and you multiply by that by the number of people that are infected, which is on the order of 1 million, and you reach this 1 kilogram value, which I guess is kind of like a fun fact. I'm not saying it has any, deep meaning, except for using these things, we can derive all the other results that I'm mentioning, that some of them I think could be of more interest. If you look at the overall mass throughout the pandemic, you need to look at how many people are infected in general. And I guess an optimistic scenario would be that we'll be ending with 500 million, and pessimistic one is about 5 billion. And you do those multiplications and you reach that overall will be dealing with about somewhere between 1 ton and 100 tons of the viruses throughout. Okay, and this is why I was running fast, because I wanted to get to this part, which is kind of like my final part about the mutations, the evolutionary implications. So first of all, if you look at the cycles of replication, you need a few cycles of replication from the beginning, where you have just a few virions, to the state in the end that you have somewhere between 1 million to 100 million infectious units. In order to achieve that, you have about 3 to 7 replication cycles. In each one, you have an amplification of 10 to 100 infectious units per cycle. And if you do that multiplication together with the mutation rate per nucleotide per cycle and the length of the genome, you get about half a mutation per lineage per infection. I would mention that this mutation rate has been measured for other beta coronaviruses and not directly for SARS-CoV-2, which is one of the gaps in the knowledge. I'll mention some more in a second. But now here's an interesting thing. If you take that and you multiply that by the number of overall infectious units produced in a person, you find that within a host, you have somewhere between 10 to the 5 to 10 to the 8 mutations per host that are being produced. And if you compare that to the possible mutations, what are the possible mutations? We know the length of the genome, we know the 3 alternative bases in each one, meaning that you have about 10 to the 5, 100,000 single bases that could, mutations that could happen. So you could say that basically in each person, through an infection, we're basically covering the whole space of potential single base pairs. And we're covering some fraction of the 2 base combinations. That's within a host. There's a huge bottleneck when you're infecting the next person, or when you're doing the infection. And from that, you can ask about what's happening through these bottlenecks as well. So as I've showed before, it's about half a mutation per infection, and there's the generation interval, the time between infections, of a few days. And from that, you get what I was mentioning before, 3 mutations per month, roughly. And if you take that over the whole population of infections throughout the world, you're getting, again, the span of variation. I won't get into all that in a second. I would mention that, as I was mentioning, if you multiply that by the number of worldwide infections per day, you're covering all single base mutations, those 100,000 appearing every day. So I think it gives us some sense of the fact that it's very hard to ensure that we won't get variants that are single base differences from existing ones. Okay, let me mention the knowledge gaps, because that's my ending slide, where I want to see if anybody has insight into that. Please reach out in one form or another. This 10 to the 4 RNA copies per infectious unit, we're really interested to know whether it's correct. We could tell you how we derived it, but we're interested to know how does it arise. So any insights on that would be interesting. And similarly about the ratio between RNA copies and viral particles. The yield of virions per infected cell, kind of like the burst size, if you like, for lytic bacteriophages, we're interested to know, again, that this is based on values that were measured for other bacterial beta coronaviruses. If you know of values for SARS-CoV-2, that would be fantastic. The mutation rate, if that has been measured or analyzed by any of you, that would be interesting. And finally, the clearance rate of virions within SARS-CoV-2. We've been using information on intercellular clearance and doing some derivations there, but that I think would help make the results more concrete. And I've added here an option if you saw anything else that you are interested in, we'll be happy to communicate, work on together, etc. I think that's what I had for you. If you're interested, then you can, and you have the connection, but I guess this would also be available online. So the people online, you're welcome to look at the current draft by looking at this QR code. Thank you very much.
Thank you both for wonderful talks. And let me, Reinhard, is there any business that we need to attend to before we go to questions?
You know, if there's time, we could talk a little bit about a suitable time for extracurricular talks like the one we had this morning by Rita Colwell. There were relatively small attendance, and probably it was because it was just an unfortunate time. So maybe we could get some feedback on what good times would be, and we can do this now, or people can just contact us by email.
Yes, this was today we had an experiment where we had an extra talk at 11, and probably two meetings of the same working group in the same day was too much. But Rita Colwell gave a wonderful talk this morning, and of course these things are recorded. I don't want to take time away though from our speakers today, so let me turn it over and open the floor to questions for two fascinating talks about how much detailed quantitative measurement we can make about viruses, and also how much we don't know about viral replication. So please, questions. And again, I encourage those who don't normally ask questions to be the first in.
I can take a minute. Just there was a question in the chat about zoonotic reservoirs. So we've been playing around with that. It also somehow cosmically connects with the census of life on Earth that we're doing for the global mammals. For example, what's the overall mass of bats in the 1,000 species of bats around the world? And I think humans dominate in the current situation, where we have about 1 million infections per day across the world. I think the reservoir in humans is much larger. Saying by how much is it larger in terms of how many orders of magnitude, that's very challenging. But if anybody's interested in discussing that more, we're interested in that as well.
John, are you raising your hand?
Yeah, wait a minute, I got a feedback problem here. The zoonotic thing is our working group is trying to build a zoonotic subgroup. And I would really like to talk to you.
I think many of us are interested in the differences and similarities between the infection of a particular virus in humans and host reservoirs. And so that would be fascinating to talk about. I wanted to maybe ask a question, both of you, because both of you showed data that are related to it, which is questions about how you understand superinfection, both in terms of whether for single virus, whether the infection in a single cell is initiated by a single virion or multiple ones, and if a single one, what do we know about how the cell then excludes subsequent infection? What data do we have about multiplicity of infection within single cells? And then potentially, specifically, John had those beautiful, very tantalizing pictures of tracking strain competition data, if any, on superinfection by multiple viruses into single cells. So either or both of you, I think, have things to say maybe to that.
Sure, I can start off. We've done most of our experiments on a rabies-like virus called VSV, vesicular stomatitis virus. It's a single-stranded RNA virus of 11 kilobases. And on multiplicity of infection, we've done infections up to 100 virus particles per cell, where you can do the quantitative PCR and find the genomes inside the cell. So what you put on the cell, you can see inside the cell. So you do 100, you do 10, you do 1, and those sorts of treatments you can find inside of the cell. And we've done things like disable cellular translation. So there's no protein synthesis, and you can just watch the viral genome after it has entered the cell. You can see the degradation rate. It just degrades. In the presence of protein synthesis, of course, you make the polymerase and you amplify that genome. So that's an example, at least, where you can, what you put in is what you also, what you put onto the cells is also what you find inside of the cells. I should say on the studies where we've done this single virus particle on single cells at low multiplicity, then, a fraction of the cells that show GFP. So this means that the virus genome gets in and it makes detectable GFP. Of those, up to 20 or 30% do not go on further to make any detectable virus particles. So you think about viruses getting in at a small one, or let's say one genome that gets in, and a sizable fraction of those that make genes that actually synthesize detectable protein, they don't go on and go all the way through the cycle to be productive. So there is probably a degradation, and some stochastic simulations suggest that, yeah, there can be degradation before you are able to amplify. This has also been found by Udo Reichel's group studying influenza, single cell infections. So that's a kind of two extremes, from one particle, one genome per cell, up to 100 genomes per cell. I'd have to think a little bit more about some of our data on the activation of innate immune responses, but we have done some on the two-color side, where we look at the viral color and we look at the host response color. I can show some further slides later on, maybe that might be of interest.
Can I jump in? What I think is a related question is, hi, this is Fred Adler. So it seems, I've gotten the impression that for some viruses, a huge fraction of produced virions are not viable. You know, either they don't have RNA or they're just not assembled properly. Can you get estimates of those? And what do they look like for coronaviruses compared to some other viruses?
Ron, do you want to take that one? I think that might have been one of your questions, actually.
Yeah, so I have it as a question, but maybe John, you have it as an answer.
I'm afraid I don't, but that's one of the burning questions for us, because I'd love to understand that 10 to the 4th. So the 10 to the 4th, you can experimentally, you can go after. I would love to see what fraction of those 10 to the 4th are actually inside particles versus free RNA. So you can treat samples with RNase, which will digest any free RNA. And generally, the packaged virus particles are protected from that RNase treatment. So if you isolate then virus particles following RNase treatment, then you can find total packaged RNA in those particles. And what you would like to do is count those particles. You can do it under the transmission electron microscopy, or you can use this kind of tunable resistive pulse sensing that we've done some work with to count. It's a kind of flow cytometry-like approach for virus particles. So that gives you particles, but then you also want to count infectious particles, and that's where you use a plaque assay. So many of you might know that standard virus textbooks like Flint in their first chapter have a table of various viruses and particle to PFU ratios that are all over the map, up to thousands or tens of thousands of particles per plaque forming units, meaning that only a small fraction of those particles that are made are actually infectious. So I don't know what the number is for SARS coronavirus. I do know that for mouse coronavirus, there have been studies in the 80s and 90s. I don't know what the numbers are on those, but they definitely found defective interfering particles on those. Yeah, I guess that's about what I can say about it. The experimental methodologies are there to figure out this, and for us, it's one of the first measures. We're starting to grow up human coronavirus now in the lab. Nan Jiang and Huicheng Shi, who are in the audience, are leading that effort. And one of the things we want to do is try to make this kind of measurement.
Could I make a comment?
Absolutely.
John, I don't know if you know Ray Pickles and Rick Boucher and Ralph Barrick at UNC.
Yeah.
But they have told me with some degree of confidence that the SARS-CoV-2 will, when it infects, say, a ciliated cell in the upper respiratory tract, that it generates 50,000 copies a day with only 4% of them infectious. So those are the bounds that they give to us for the modeling that we do.
That's great. Is that copies of RNA or is that copies of particle? Are those particles?
I don't know the answer to that question. What they're saying is that 2,000 infectious virions a day for 3 to 5 days per infected cell.
That's great to know. That's definitely a group I would trust. So yeah, that's great. Have they published that?
I doubt it.
Okay. All right.
There's a paper by Barrick and Boucher in Cell last year that will have as much data as they published at that point, but I don't know about any since then.
I appreciate that. Thanks very much. I had a question actually for Ron. You focused a lot on the mutation rates, and for the coronaviruses, it has this exoribonuclease, which seems to cause extensive recombination. So I'm just wondering, my sense is, so this is from the Denison group, which is also, I think, very well established in the coronavirus area, that they find extensive defective or genomes that are lacking many essential genes, which might be a signature of potential defective particles. I wonder if you've thought about trying to estimate sorts of maybe some of the numbers. I guess that 10 to the 4th is probably based on not full genome sequencing, but maybe PCRs on a particular gene, in which case it's conceivable that these could be defective genomes that are generated by the exoribonuclease. Have you looked into that aspect, the recombination aspect on the coronaviruses at all?
Thanks. We have not looked into that specifically. And now that you mentioned that, we will, and we could maybe afterwards and see maybe we could explore that further.
Yeah.
This is a great set of questions.
Yeah, this is Alan Carlson. Can I make a few comments?
Go ahead, Alan, please go ahead.
Yeah,
Nice
so this a couple of comments. So one on this particle to infectious particle ratio, that's been looked at, in HIV and SIV, again by doing PCR to count virions and then doing TCID 50 or plaque forming units, and also by diluting serum after you've done PCR to multiple folds and seeing if you have infectious units, at least in animal models. One of the things that's been noticed there is that very early in primary infection, the ratio of infectious particles to total is very high. And that with time of infection, even a few weeks or even a week or two, that ratio goes down. And it's maybe correlated with making antibody responses and generating neutralizing antibody or just having autologous antibodies, that's binding to the virions. At least in one set of experiments using SIV, if they took the plasma, and they asked whether or not if you change temperature, for example, is this property heat stable or not, which they were using temperatures at which immunoglobulins would be stable. It seemed to be heat stable. There are other plasma proteins that also, at least in HIV, people have thought may be involved in blocking infection just by binding to the surface of virions. So one of the concerns, at least in SARS, since most of the samples one gets is after people become symptomatic and possibly even after they're hospitalized, we could be looking at samples that are 2 weeks into the infection or so, at which point at least some of these effects may be relevant, in addition to all the genetic effects that go on. The other thing, Ron, in terms of about the virus being produced, at least looking at clinical samples from patients who've been hospitalized and measuring the viral loads, near the time of admission, there's a huge variation in that. One of my collaborators, Jeremy Gage, has a paper last month in PNAS where they looked at 655 hospitalized patients and have viral load data in that paper. It's NEONT, I'll send you the reference. And the data on admission, if you look at their plots, go from a few hundred copies per ml, and these are nasal pharyngeal swabs, up to almost 10 to the 12th per ml. And they do have patients up there, 10 to the 10th, 10 to the 11th, 10 to the 12th, that sort of peak viral load. If you take a median or so, it's probably more like 10 to the 8th, but there are examples of very high viral loads. And again, they may have missed the peak when they've gotten them. So that's per ml on a swab, not total lung or anything else. So I'm a little skeptical, at least of the maximum amount of virus that can be produced in a person. I'll send you that paper. And there's some others also that have very high ratios. I've also seen and done calculations on this 10 to the 4th between infectious and non-infectious, just on papers that have been published where they give RNA and they also give plaque forming units. And I've corresponded with some people who've done work in non-human primates, and something like that they've measured also in their stock viruses.
Let me ask you, Alan,
Sure.
or others in the audience here who might have a better sense here. You mentioned the viral load, estimating it by the PCR, which I guess the question for me is that, I guess the distinction is here, free RNA versus particle encapsulated and protected RNA. So viral load, yeah, it's easy to do PCR, so you can quantify a number of RNA strands very nicely. But if you're quantifying those from a patient sample, then they might be free in solution or released by broken lysed cells rather than actually packaged into virus particles. And I think in general, people don't go to the trouble of trying to find the particles, especially if they're non-infectious. But I wonder what fraction of actual RNAs get packaged, and then what fraction of those packaged ones.
Yeah, I don't know that answer. I mean, you mentioned that before, and I think it's be a nice experiment to do. It's not that difficult for a lab to do the RNAs first, but I don't know of anybody who's done that.
Yeah. Okay. Good.
I know the other thing that people have been concerned with, and they're taking clinical samples, which are usually swabs, and doing PCR, that they may be measuring a virus that's actually in cells, so that when they do those swabs, they're not only getting the fluid, but they're also swabbing off cells.
Yeah.
And some groups have started also doing PCR on host genes to try and get some idea of how many cells they've isolated in that sample, and whether they're looking again at intracellular virus that's being isolated in their PCR procedures, where they do have to break open the membranes, either the viral or the cell membranes.
Yep. Okay. Good. Thanks.
Another confounding issue.
Yeah.
So sorry, let me break in for just a second. We're at the canonical hour, so the official part of the meeting is over, but that we have a half an hour to discuss. Given the intensive discussion, I don't know, Ron and John, would you prefer to be in breakout rooms so that you could talk to people separately, or is it more productive to stay in the main room and have a joint discussion? It's really up to you either way. If you feel that you could play off each other effectively, then maybe it's better for everybody to stay in the main room to talk. If you think it would be better to be able to talk individually, I'll put you into breakout rooms.
I personally doubt that I'll have the opportunity to visit Madison anytime soon and talk with John. So I think staying here in the same location is actually great for
That's fine. So then
the same goes for me going to Israel. So right.
Okay.
This just seems to fit together too well.
So then there's no need for the breakout room usage. We'll just stay in the main meeting, and we can talk in a friendly way for the next half hour. So please.
I had just one other comment for Ron, and in addition to measuring peak viral loads, or doing that for your estimates, why not do the area under the viral load curve that you would get by studying infections in either humans or in non-human primates, and get some idea of the total amount of virus being produced during an infection. And again, that's complicated because we don't know if it's all infectious, and many of the clinicians think after a week, 10 days that the viruses that you're measuring by PCR are probably not infectious, and there are a few studies about that. But at least you could do the area under the curve for some period of time to get something a little bit more physiologically based. And also then when you're going into calculating,
yes, so I think that
possibly go on. I've done and published some calculations like that for influenza, where we were interested in the amount of virus being produced and the possibility of mutation. And the idea was to calculate the actual number of virus particles produced over the course of the acute infection, with the understanding that every viral particle is carrying an RNA, and each RNA, there's some mutation rate in producing that RNA. So anyway, we based it on the area under the curve, and then also tried correcting for the fact that what you're measuring is only a subset of the virus produced, because it's also simultaneously being cleared, at least in the in vivo situation. So you got to do a little calculation as to total production, which is slightly different than what the area under the curve.
Yeah. So I will see if maybe one of the co-authors wants to also mention on that. But I think what you're suggesting is the more rigorous way to do it. Mm-hmm. The fact that we don't have the kind of developed models that we know your group has been developing through that makes it harder for us to do that, because it's much easier to us to do what we used to, like in this Fermi problem spirit, to do the multiplication and divisions. But if you really want to do the area under the curve properly, you need to have good models for that, which we don't. So it's really a question of that, and maybe we should do better.
Yeah,
especially if you think that this
you need to collaborate, or I can just send you some references and make some suggestions as to how you might improve it, or at least at least recognize that it's a limitation of what you're doing so far.
I'll just say that we are currently using a kind of a crude version of the models that you're using, and it's basically highly dependent on the clearance rate of the virus, right? If the clearance rate is very high, then the number would be much higher than when it's low. So I'm not completely sure how well constrained are the estimates for the clearance rate based on the models. how sensitive are they to other model parameters?
Yeah, there are some estimates that people have published, and we've published based on trying to fit clinical data. But we know when the clearance rate is sort of the fastest time scale, which is we know that's true for a lot of other viruses, then it becomes very insensitive. Typically, for human viruses, for HIV and hepatitis C, we've got minimal estimates that their half-lives are something like 45 minutes in circulation. And that's a very good estimate for C. There's estimates in non-human primates where you can infuse labeled virus or single cycle virus and sample very frequently. And there we've gotten clearance rates of order 5 minutes, half-lives of 5 minutes or less. There have been experiments again in non-human primates where you infuse known virus at known rates and try to get a steady, wait till they attain a steady state. And then you know the clearance rate out of the circulation is equal to the rate that you're putting it in. And also that comes out on timescales of minutes again for a variety of viruses. And so from clinical data, it's impossible really to get those types of clearance rates. And you're right, if they're very, very fast, then the total amount of virus being produced would be sensitive to that. But,
Alan, correct me if I'm wrong, but there's also the issue of the clearance rate out of the cell, like in the body, extracellular, and there's the issue of the clearance rate intracellularly.
Right.
And I think the numbers that you mentioned were talking about the clearance from the body, meaning extracellularly.
Yes, it's extracellular.
And I think for many of the things we're interested in the.
Yeah, and actually the numbers I was mentioning are the ones that are easy to get, and that's clearance rate out of the circulation, and whether or not the virus is actually being destroyed in tissue, or some of it can be, especially if you're studying acute infection, some of that can just be redistribution. Until you sort of fill up tissue binding sites. So it's a little unclear exactly what that clearance is body-wide and what those rates are. From the easiest thing to measure and what is measured is out of the circulation.
But is there anything measured about the intercellular clearance rate? How fast produced virions are getting emitted into the extracellular?
Yeah, very few estimates of that. We were looking at making complete models of our life cycle, like John does for hepatitis C. And our best estimates there was that there was extremely slow, if not no clearance within the cell. And there are some molecular immunologists, somebody like Ralph Bartenschlager, the University of Heidelberg is a real experimental expert, and he sort of concurred with us that they don't see much degradation intracellularly, at least of hepatitis C. It's also a secreted virus, so same as SARS coronavirus, but I don't know very many measurements of that. It's something that you could do in vitro. John could possibly do that for the viruses he's looked at.
We haven't worked with hepatitis C, but we've got our hands full now starting on corona.
Yeah, no, it'd be interesting to look at, viruses that are, assembled inside and go out through the secretory pathways.
Yeah.
Our feeling was that once you, assemble a virion, it's highly unlikely that the cell would degrade it rather than just export it.
Yeah.
I mean, the RNAs intracellular, or the DNA intracellular
I guess
could be degraded, but once you make a viral particle, I think the degradation is unlikely to be very, very rapid or frequent.
Yeah, I might not be using the right term, but actually I would be curious to know if you, John, or anybody else I'm not talking about the degradation of a virion inside the cell, but I'm talking about the rate at which they're being secreted. Like for how long is there the residence time of a virion inside the host cell before it's being excreted?
Yeah.
I guess then, so you would like to, you're saying that in principle, you can break open the cell and you can find infectious virions before it's secreted.
Mm-hmm.
That is possible, at least I know for phage systems that we've worked with. Phage T7 is a double-stranded DNA virus that you can break open the cells before they would otherwise lyse,
Mhm.
and then you can capture infectious particles. So they are assembling within the cell and infectious prior to cell lysis. In other viruses here, we've got the viruses that are being secreted from the cell surface. Then they're not really virus particles until they have left the cell, in which case they're taking along some cell membrane. So I guess one couldn't really define a virus particle in that sense until it has been separated from the cell.
Yeah. In the SARS coronavirus 2, I don't know if you've seen the transmission electron micrographs of it. I mean, they seem to fill up intracellular vacuoles, and they pretty much look like intact virus particles that are then being secreted.
Okay.
That's my understanding of it.
Do you know if people have isolated infectious virus particles from those vesicles?
I don't know.
Yeah, I know they're replication centers, but I wasn't aware of people. My sense is that they're probably very fragile, and it might be hard to do that, but it'd be interesting.
Yes.
So, I mean, one of the things that you both alluded to, there are several people in the room who do multicellular modeling. So instead of doing population level within the tissue viral loads, they're trying to model individual cells and their effects. And you had some nice, both of you had some nice statistics on variation in burst size between cells. One thing you didn't talk about, although you alluded to it, was the lifetime of the cell after infection. Something else one would love to have is the kind of thing, for example, that Jim Fetter would calculate, but or last week David Oda, which is the rate of viral production as a function of time after infection for a single cell.
Mm-hmm.
And I wondered if you could talk a little bit about what level of single cell data is possible. It may be that you don't need these single cell models, that the aggregates are enough, but there are certainly people in the room who would like to know, if you're going to model individual cells in the lung tissue, what kind of viral model should we have inside of each one, and how do we calibrate it?
You want to take that, Ron, or I'm actually looking for, so we did some work on single cell kinetics. And this is where you essentially dilute down you infect cells at high multiplicity. So these are not single virus particle infecting single cells. This is many virus particle infecting small number of cells so that all the cells get infected. And then it's to put those single cells in their own little wells with their own supernatant and to sample them periodically. So it's a crazy experiment, but I had a very dedicated graduate student, Andrea Tim, who had very good hands, and she did this. So let me see if, I'm not sure if I can share.
You should be able to screen share.
Okay. Yeah, so this is the paper from 2012. And so she was actually sampling multiple times supernatant from individual cells and replacing the supernatant with warm medium, and then allowing it to sit around longer and then sampling again. So she would get these, yeah, I guess this is the experimental setup in A. You make sure you have high multiplicity, so every cell you get is essentially infected. You dilute down, then you look for individual cells and individual wells, and then you sample and you replenish supernatants, and you generate plaque counts as a function of time from individual cells, and essentially integrating then the counts. This is your area under the curve, Alan, except for it's a single cell. Mm-hmm. Then you can get a total virus per time. So when she did that for, I don't know, this number of cells, 12 cells here, then in A, those are the single cell counts as a function of time. then so that's PFU per hour, you could say. And then you integrate those and you generate these single cell one-step growth curves, which are all over the map with respect to yield, because we've got some that, here's one down here that's maybe 20 or 30 yield. And then here's one that's up around 10,000. These are all cells from the same dish. What's interesting, I think, also here is that the time is relatively narrow when these single cells are producing. The window at which they are producing is between maybe 5 and 8 hours, or maybe 5 to 10 hours. So huge differences in yields from single cells, but relatively narrow window in which they're producing. I think we did some additional analysis here. So you can look at various correlates here of rise times and latent times on this. So the longer the latent time or that you're waiting around, the higher the rise rate also tends to be. So I'm trying to remember going back to so we have these single cell data. I guess I'm now trying to revisit what was the burning question that we wanted to answer.
Well, again, I was saying for us, we have models of viral replication inside individual cells. We have a lawn of epithelial cells in our simulations.
Yep.
So we'd like to be able to, the aggregated data on net viral load in the simulation is not very informative about the microscopic parameters and variation between cells.
Yes.
And so, and if you're thinking about trying to explore therapies, that microvariation may be very important, and so you'd want to get it right.
Yep.
It's like trying to measure a tumor by looking at total tumor mass.
Yep.
It really is not a sufficient. The total viral load isn't sufficiently informative, perhaps, to be able to really. I mean, you could certainly build useful models, and Alan builds wonderful models that way. But if you want to do models where you model individual cells, then we need to be able to try to understand the survival time of the cell after infection, the production, all of these questions that Alan was addressing.
Yeah.
There's something else that you alluded to that came up. I heard a very nice talk by a Polish group that was looking at spatial correlations in they were using a different not a coronavirus. And you were looking at interferon marker, a vital interferon marker reporter with a viral reporter.
Yeah.
They were looking at correlations between interferon production levels, both RIG-I and STAT-P.
Yeah.
And they were different. Those reporters were very different. And the spatial correlations between infected cells, related viral production, and those two interferon markers. And they had rather complicated spatial patterns that were not what you would simply expect from a classic plaque assay where you have interferon getting ahead of the plaque.
Yeah.
They were getting very complicated, almost for a physicist, antiferromagnetic patterns where you get alternating high-low patterns.
Uh-huh.
And I wondered what you were seeing if you were doing, and Ron, I don't know if you're doing those kind of spatial analyses, but when you look microscopically at spatial correlations in those kind of two-factor visualizations, what do you get?
Actually, I don't think I've seen anything like high-low, as you mentioned, antiferromagnetic, but it does remind me of the idea of potentially having Turing patterns. So, you know, when you have a diffusible activator and inhibitor, and the inhibitor has a higher diffusivity, then you can be generating dots or stripes and generating these spatial patterns in essence. And I wonder if that might have been happening because we have diffusible virus particles and diffusible interferons. And the interferon, the signaling molecules generally are smaller and faster to diffuse. So maybe there's something like that happening.
What they were seeing was actually, I think they felt I'm sure these things are very specific to the virus and also certainly to the cell type you're using in vitro. But I think what they were seeing was typically that and again, it was particular virus. The virus had different ways of shutting down the two sides of the interferon pathway. And so depending on the race condition between viral proteins, particular viral proteins, and the two interferon pathways, you could get four states, for example, where one of the interferon path, the paracrine signaling was happening, but not the internal, and vice versa. So you had RIG-I versus the STAT happening independently of each other. And it was also affecting the way the virus was being shut down. And so I guess my point was simply that I think understanding that the levels was fascinating. What you showed at the single cell was fascinating, but looking at the spatial correlations would also be really interesting. And for the kind of spatial modeling a lot of people here do, it could be very interesting to try to replicate that.
Yep. Just the variability, one aspect here is the, moving away from ODEs to stochastic models, I think is probably something that is an approach that many have done also. I think Jacob might have had a question, is that?
Joy, I'm sorry, I didn't mean to monopolize the conversation. Go ahead, Jacob.
I had a question, I put in the link. Someone answered my question. I just noticed it, but I put in a link and I'm looking at what you're doing, and I'm not in that field. I do micro simulation, but like population models. But I'm trying to figure out what you're doing different than what I see is going on. So John Rice sent me this paper 6 months ago, something like that, long time ago. And I was looking at it, and this is actually this is for you, maybe because the people that are mostly from Israel, you might know them. What's the difference between what they're doing, which is actually also showing different strains of viruses, and what you are doing? Because you're also saying there's a number of viruses and you're also counting them. What's the difference? Is it technique, or are you doing the same, or what's going on there? Just have a glance.
So I referred to that. I referred to that in my answer in the chat. But it's a very good paper and it's a very good group. We're in touch with them very a lot. And I would say what they did. We're doing different things in the sense that they're doing the hard work of really taking the samples, doing the sequencing, building the phylogenetic trees. We're doing the easy part of just taking some numbers and doing the multiplications and divisions and checking if things fit together. So they're doing an important empirical case study, and we're taking the more, general back of the envelope calculation, seeing how things fit together and what we could generalize from that. So that's the overall general.
You're doing the higher level modeling. This is what you're telling me.
Yeah, maybe you could say that way.
Okay, this is your base data, what they're doing, or you have your own data?
No, so I've basically shown you the data that we're having. So we just it's not the same data in the sense that we started from very fundamental numbers, but then what we did is verify that the results that we're getting are in line with the results that they're getting. For example, the results that they're getting are also the result of selection. So it's not only neutral. So there's fine points here about what are you taking into account, only the natural drift or also the selection pressure. Necessarily in the real world, there's also the selection pressure, but often you find that this is, how much does that change the order of magnitude of what you're doing? Often it does not in terms of those things.
You said that you are connected to them. Do you think that what they're doing will be interesting to this working group?
I hope so. Yeah. So the last author is there.
I tried to contact them multiple times without success. Really, to like all the emails. I looked up at all the emails. If you can, then can you connect them to James Glazier here?
Sure.
Thank you. Thank you. Then I'll be able to ask them the questions.
Okay. I see, Jim Fetter's on the call, Veronica's on the call. Do you have anything, maybe you talked to our speakers already without the meeting, but it's a good opportunity given to put some of the questions into the public if you'd like. I'm not trying to exclude anybody, but I just see Jim and Veronica as people who definitely work in this area.
Say hello. Though the very interesting discussion, we've been grappling with many of these questions too, obviously, and especially about this issue of how many infectious virions per virion, and the estimates that we've seen from our collaborators in line with this 10,000 number. But it is a little bit, it's mystifying, you know, and I've seen there's some nice modeling work that suggests that that these non-infectious virus particles can, you know, have a significant effect on the kinetics of an infection. That was work that Kevin James put out recently, his group, and this is very interesting to consider. But yeah, they're all. I actually did have one question for Ron, which was that about this issue. We haven't talked about it yet. The idea that, you know, 1 in 100,000 to 10 million lung cells being infected simultaneously, that seems really low to me, and it's very low. So we've so far only modeled infections in cell culture. So, you know, their you know, MOIs are very high, and certainly not. If we told our collaborator to do a 1 in 10 to the 5 MOI, they would laugh at us, right? So how does this? How do you view that outcome?
So I view it with fascination, and if you can prove and if you can prove us wrong, please do it now before the paper is out. I'll be happy to learn about it now.
Can I ask for a clarification on that? When you say 1 in 10 to the 5th cells, is that in an infected patch, or is that over the whole lung? In other words, if I look at a plaque in the lung and you tell me 1 in 10 to the 5th cells is infected, then I'm nervous. If you tell me that 1 in 10 to the 5th 1 centimeter patches is infected, then I'm not nervous at all, because the lesions are localized.
So what we calculate is overall. So we don't have a spatial model. So in that sense, it's like overall in the lung. And I'm guessing there's a lot of heterogeneity. So you're probably right. It's like in patches. But whether it makes sense that out of all the patches, you'll only have, like, say, 10 to the -5, that I don't have a good intuition about that. I would mention that this value does become somewhat lower or higher, depending on how you want to define it, when you take into account only the fraction of lung cells that are expressing the, what is it, like the ACE2 and the TMPSS or whatever it is. So we've been taking that into account, and then it goes from 10 to the 5 to 10 to the 3, or 10 to the -3, if you like. Somewhere between, instead of 10 to the -5 to 10 to the -7 to 10 to the -3 to 10 to the -5. So still you need all this heterogeneity and patches and everything, but the values are somewhat different.
I mean, in the spatial models we do, patch to patch, because we're simulating patches of a few thousand cells, there's a huge amount of heterogeneity. At certain level, even for the same parameters, some patches you'll get a few cells infected and the infection doesn't spread. Other ones, every cell is infected. Other ones, some fraction of the cells are infected. Now we tend to work, because we don't want to have to do that many replicas, with parameters where most of the cells get infected most of the time. But if you move the parameters just a little bit, you could wind up with a situation where 1 in 1,000 patches had a large rate of infection. Wouldn't have to move the parameters very much to get there.
Hi, I also have one question. So there are more and more papers appeared that showing that this SARS-CoV-2 could also productively infect human gut and enterocytes. So did you see any estimation? Because it's look like the virus production from those cells might be kind of much less, almost like go under the radar of the immune system. And that's what might even make them more like persistent for a while. Do you know anything about it?
Yes, so I don't know if that answers your question, but if you look at what I've just shared here, so in the digestive system, even though the overall mass is large, kind of like the lungs, the values that we found reported for the number of RNA copies or infectious units is close to the detection limit. So even though it exists there and you can find them in, say, in PCRs, etc., if you do the multiplication, you're orders of magnitude away. So we think it's a small contribution in the absolute values. That
So
being
just
said, I'll mention again, this is based on rhesus monkeys. So if something is very different in the digestive tract of people in terms of the concentrations, maybe these results would also be different.
Okay, so it may be that they contribute to persistence by generated very small amount of virus, basically. Yeah, okay. Thank you.
Okay. Well, I think it's been a it's our most successful discussion session so far. So I think we have to have, maybe we need Alan, Ron, and John back to have maybe a panel discussion to promote these conversations. Are there any last questions before we thank our speakers again and let them get back to their work? No, well, thank you both, and thank you, Alan, for coming.
Oh, you're welcome.
Really appreciate it. Thank you, all of you, and we really do appreciate your taking the time to speak to us and for the great discussion. And I
Thanks
hope we'll
for
have
great
a chance.
questions. Appreciate all the questions.
Okay, well, with that, I will call the meeting to a close, and I look forward to seeing people next week, and suggestions for speakers are always welcome.